Hello student, good morning. We continue the lecture, that is secondary structure of proteins. We have to study the last period primary structure of proteins, and in this lecture we will start the secondary structure of proteins. The secondary structure of proteins is determined by the primary structure. It is the lowest energy state for a particular order of amino acids. In a particular environment, most of the secondary structure is formed at the time the proteins is synthesized and remains the same even if modification occurs to the amino acids or if segments of the proteins are removed. Proteins without a regular or repeating structures are considered random coils, an unfortunate term as the natural nature of hydrophobic and hydrophilic amino acids will make the arrangement definitely non-random. Arrangement definitely non-random. A helix. There are multiple possible secondary structures, but two occurs frequently enough that you should know their characteristics. The first of these is the A helix. The A helix of DNA, the protein A helix is right-handed and left-handed helices are possible, but the right-handed one happens to be more stable for the naturally occurring L amino acids. The protein's A helix has 3.6 amino acids per turn, with the R group of the amino acids extended outward perpendicular to the axis of the helix. The structure is held together by hydrogen bonds between the carboxyl of one peptide bond and the NH of the amino acids 4 residues above 18. The helix, because every peptide bond except those at the ends is involved in two hydrogen bonds, the A helix is very stable structure. The A helix is most stable when the primary structure contains amino acids with uncharged side chains: alanine, leucine, phenylalanine, tyrosine, tryptophan, cysteine, methionine, histidine, asparagine, glutamine, and valine. The A helix is destabilized by amino acids with bulky or charged side chains such as serine, isoleucine, threonine, glutamic and aspartic acids, lysine and arginine, the only amino acids that cannot be accommodated in an helix is proline due to the rigidity and bond angles of its structures. If an helix exists in proteins, it will stop when proline residue is encountered in the primary structure. A keratin, the major protein of skin, hair, nails, and some of the intracellular filaments, is predominantly in the form of an helix. Several of these helices are twisted together to form long fibers or ropes. The tertiary structures of a keratin is stabilized by disulfide bonds between adjacent helices. Because the individual helices are held together by hydrogen bonds, a keratin can be denatured by detergent, heat, and other agents that disrupt hydrogen bonds. When this happens, the molecules can be stretched out considerably longer than its originally length. To see this change for yourself, stretch out a wet strand of hair. Permanent waves disrupt the disulfide bond between helices. When the bonds reform at new location, the shape of permanently waves hair is changed and curl results. B structure. A keratin we have to study, and second is B structure. The secondary of the important secondary structures is the P structure, or related pleated sheet. In this structure, the protein chains are extended with the R groups alternating above and below the backbone. Hydrogen bonding occurs between the peptide bonds on chain running parallel or anti-parallel. These bonds can be between different chains or the same chain folded back on. Results, the hydrogen bonds in the B structures are oriented perpendicular to the direction of the protein's backbone. As with the A helix, all peptide bonds can be involved in hydrogen bonding because of steric interactions between R groups. The B structure can form only when the primary structure contains amino acids with small side chains. B keratin found in silk and the beaks of birds is arranged almost entirely in a pleated sheet structure. It does not changes conformation when wet or when heated at low temperature. Because B keratin contains a cysteine, it has no disulfide bond and is not altered by treatment with disulfide reducing agents. The preponderance of amino acids that have small R groups allows the beta keratin to assume the beta structure. Okay, this is the secondary structure of protein. Secondary structure of proteins. We have to study the primary structure and secondary structure of protein. Okay, thank you. Okay, thank you. Primary structure and secondary structure we have to study here in this lecture. Okay.